is in December 2019, severe acute respiratory syndrome coronavirus 2 SARS-CoV-2 was identified in Wuhan, China, which spread rapidly and has become a worldwide public health challenge. And this is the structure of the coronavirus, and this is the spikes, and this is the genome which is present inside it. So the first line of defense mechanism against this COVID-19 virus is the innate immunity, which is the principle of any infection. The first line of defense is the innate immunity, and the body responds to this viral infection immediately with non-specific innate immune response, which is include NK cells, macrophage, neutrophils, and dendritic cells. And these cells slow the progress of the virus. And first site of the infection is the upper respiratory tract. And these innate immune cells may even prevent it from causing symptoms. And early control of the viral replication by type 1 interferons and complement proteins and other innate immune mediators limit viral spread within the host during the early phase of the disease. So this is all the innate immunity, which is prevent the viral replication. And if this innate immunity fails, so the adaptive immunity to COVID will be stimulated, and this involves cells which is called B cells that produce antibodies, and nearly everyone infected with COVID-19 makes antibodies against this new coronavirus within 1 to 2 weeks. And the first antibody will be produced, the IgM, which is, as a principle, it is a primary immune response in the IgM production and indicate recent infection. And later on, IgG will be developed, which is indicates secondary immune response, indicate either past infection or recovery. And these extracted antibodies from COVID-19 survivors and also found those antibodies were neutralizing antibodies. As we know, the function of the antibody is a neutralization. So they found that these antibodies had a neutralizing function that's capable of binding to the virus and its block entry into the host cell. And this figure shows us the antibody production with relation with the infection. And this is here the window phase, which is the asymptomatic stage, and the person is asymptomatic, but he's a carrier of the virus, which is very important because he spread the virus and shed the virus, and he while he was asymptomatic, and it's called window period. And this is the patient's infection, and here there is no antibody detection, and only method to detect the infection by the real-time PCR detect the genome of the virus in the blood. And after that, the infection will be appear here, this zone, and the IgM will be detected. And here the IgM reach its peak and then will be decline. And then the patient will be recovered. And then the IgG will be increase in its titer and then reach its upper limit. And here the patient in the convalescence. And then the IgG will be decreased in its titer. And here we can, in this period, in the early, in the window period, we can detect the infection by the PCR, by the real-time PCR detection, the presence of the viral genome. And then here in the symptomatic will be detect the IgM. And then when there is, when the patients will be recovered, decrease in the IgM and increase in the IgG. And in the convalescence period here in this zone only will be detected the IgG. And this is the best figure to explain the antibody titers during the infection. And we detect the COVID-19 by the PCR by taking the nasopharyngeal swab, and we use the PCR method, as you know, previously using the primers of the virus, and then amplify the genetic material of the virus, and then we detect it by the real-time PCR using the fluorescence markers found to the copies during PCR are released and can be detected when amplification occurs. And the positive results when there is viral genetic material in the sample after amplification and releasing enough fluorescent markers. Here we can see the increase in the curve, the increase in the amount of the fluorescein, which is indicate positive. While negative, there is no viral genetic material in the sample amplification and will not occur, and no fluorescence marker will be detected here as this dotted red one. And the another cell in the adaptive immunity that attack the COVID virus, T helper, which is involved in the defense mechanism, help the cells to make antibodies, and also T cytotoxic and over-exuberant response in some patients cause inflammation that block the airways and high level of the cytokine release, which is called cytokine storm, which is type 3 hypersensitivity. And this is the same what's happened in the rheumatoid arthritis, which is called the cytokine soup, while here in the COVID, which is called cytokine storm. And it is type 3 hypersensitivity reaction because it's associated with increase in the neutrophil count and decrease in the T lymphocytes with microthrombi. Sorry, with microthrombi formation that obstruct small blood vessels, including lungs, kidney, liver, heart, leading to multi-organ failure and death. And this is the immune mechanism to COVID virus, and the COVID also had its own evade mechanism from the immune response. So the virus encodes specific gene products that antagonize both the innate and acquired arms of the immune response. This is first. Second, enhance the production of antibody-dependent enhancement, ADE, that facilitate the dissemination and entrance of the virus inside the cell through spikes binding ACE, angiotensin converting enzyme 2 receptor. And the third one, the virus had high rate of mutation. So we have three types of this virus: the type A, type B, type C, and this is occur due to the mutation that's occurred in the virus to evade the host immune defense mechanism. Now we have what's called herd immunity that some countries applicate this principle in order to overcome this pandemic. And the herd immunity, also called herd effect, community immunity, population immunity, or social immunity. As you know, the most important method to prevent pandemic COVID is social isolation and social isolation and distance between each two persons about 2 meters. While here in the herd immunity, all the population did not quarantine and did not isolate them, and this is a form of indirect protection from infectious disease that occurs when a large percent of a population has become immune to an infection, thereby providing a measure of protection for individuals who are not immune. And here we have the herd immunity, how it's worked, and here we percent vaccinated, and this is the percent vaccinated 75. So the here percent vaccinated 25, percent vaccinated 30. and here percent vaccinated 50 and percent vaccinated 95, and this is the small one which is get immunity from this yellow one which is vaccinated or infected with the virus. Now we have the bacterial infection. Bacterial infection, we have two types of bacteria, intracellular and extracellular bacterial infection. Now we will deal with the extracellular bacteria. And the immune response to extracellular bacteria is secretory IgA, which is a blocked attachment of the bacteria. And important, the humoral immunity. It's a principle. Each extracellular, it is a humoral immunity. Each intracellular is cell-mediated immunity. So we will deal with extracellular bacteria. So the main immune response is a humoral immunity, which is responsible for production antibodies that remove bacteria, neutralize its toxin, activated complement, and with lysis and C3b production that enhance the phagocytosis, and also production C3a, C4A, which is act as anaphylaxins, mediate mast cell degranulation. And while the immune response to intracellular bacteria, it is cell-mediated immunity. When the innate immunity, which is NK cell, which is the first defense mechanism, and if the innate immunity fails, so the cell-mediated immunity will be stimulated, which is especially the delayed hypersensitivity reaction, and secretion of gamma interferon from the CD4 cells and activate the macrophage and enhance the phagocytosis. And also bacteria had evade mechanism through secretion of a protease, which is a clear secretory IgA, and the best example, Neisseria gonorrhoeae, and second antigenic variation that the bacteria change its cell surface structure like the Pili of Neisseria gonorrhoeae, and some bacteria have polysaccharide capsule which inhibit phagocytosis, and other bacteria gram-positive bacteria resist the complement mediated lysis, and other secrete the enzyme like elastase that inactivate C3a, C5a, like for example Pseudomonas, which is highly resistant to antibiotic, and most important bacteria in burns, and other bacteria secrete hyaluronidase enzyme that enhance bacterial invasiveness through the conductive tissue and through the soft tissue of the body. Another type of the infection, parasitic infection, and the type and effectiveness of the immune response to protozoa. As you know, the parasite, we have the protozoa and the elements. Here the protozoa and this immune response depend on the location of the parasite and life cycle stage of the parasite. When the protozoa spend part of their time free within the bloodstream, so the immune response is the humoral immunity. While the another stage when the parasite intracellular, so the cell-mediated immunity. For example, Plasmodium that causes malaria and African trypanosoma, which is covered with variable surface glycoprotein due to genetic variation that's called its that's caused for its cell surface so can escape the immune system, and the leishmania, which lives in the phagosome of the macrophage